Good morning, students. Today our topic is double fertilization. We have done it earlier, but the next topic is dependent on this. So I'm just doing a recap. Double fertilization. After entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. And this forms zygote, and then it turns into embryo. And the second male nucleus, which unites with the polar nuclei present in the central cell, and then fusion of three haploid nuclei is called triple fusion. And both these processes, syngamy and triple fusion, are called double fertilization. Now, what happens to the triple fusion? Triple fusion in triple fusion, and the central cell after triple fusion becomes primary endosperm cell and develops into endosperm, while zygote develops into the embryo. We have done it earlier. Now, zygote gives rise to proembryo and subsequently to globular, heart-shaped and mature embryo. From the zygote, when it's formed proembryo, then suspensor cell is given out, and from the suspensor cell, hostorium is made, and that hostorium gives rise to plumule, radicle, or different types of globular, heart-shaped and mature embryo in different species. So in this slide, we'll study about endosperm, structure, types, and development, which includes post-fertilization structure and events. So what is endosperm? Endosperm is basically name of the food-laden tissue, which is meant for nourishing the embryo in seed plants, which therefore develop before the development of embryo. So in angiosperms, endosperm is a special tissue which is formed as a result of vegetative fertilization. So triple fusion or fusion of male gamete with the diploid secondary nucleus of the central cell. So depending upon the mode of formation of endosperm, angiospermic endosperm is divided into three types. We'll study about two types in our chapter. First is nuclear endosperm. So in nuclear endosperm, the primary endosperm nucleus divides repeatedly without cytokinesis, means without wall formation. A large number of free nuclei are formed. Central vacuole appears in the central cell and pushes the cytoplasm containing the nuclei to the periphery. It is a common example is coconut milk. In coconut milk, we are having large number of nuclei, but no cytokinesis takes place. So it contains, it has very, coconut milk has very minerals because nutritive value is very high, and it's isotonic to our body. Next is cellular endosperm. Cellular endosperm is basically, which is the white kernel of coconut, which we are eating, just like we are eating the giri of it. And this is after nuclear division, subsequently cell wall formation takes place, and endosperm which becomes cellular. So it will be cellular. Now, depending upon the types of endosperm consumption, fate of endosperm. On the fate of endosperm, seeds are divided into two types: endospermic or albuminous seeds and exalbuminous or non-endospermic seeds. So the endosperm in albuminous seeds, the endosperm may persist in the seed when the latter is called when these are called albuminous, just like castor, cereals, coconut, etc. In some other seeds, when endosperm is completely absorbed by the growing embryo and the food reserve gets stored in the cotyledon, such seeds are called non-endospermic or exalbuminous. For example, pea, bean, sunflower, etc. In this slide, we'll study the stages of development of monocotyledonous embryo. The embryo of monocotyledons have only one cotyledon. In grass family, Gameaceae, these cotyledons are scutellum. We're watching in the slide scutellum. It is situated towards lateral side of the embryonal axis. The axis at its lower end has radicle and root cap attached in a sheet-like structure that is called coryza. And the part of axis above the level of attachment of scutellum is called epicotyle. It has shoot apex and few leaf primordium, and in a foliar structure called coleoptile. Epiblast represents the epiblast you're watching in the diagram. Epiblast basically represents the rudiments of second cotyledon, which were not present now. A typical dicotyledonous embryo consists of embryonal axis and two cotyledons. The part of embryonal axis above the level of cotyledon is called epicotyle. It terminates with the stem tip called plumule, which is a future shoot, and the part below the level of cotyledon is called hypocotyle, which terminates in the root tip called radicle, which forms a future root. The root tip is always covered by a root cap. Now in dicot embryo, this terminal cell produces embryo except the radicle, and plumule is terminal and lies in between the two elongated cotyledons. So this is all about dicotyledonous embryo and formation of monocotyledonous embryo.